So we are going to cut in just a couple minutes early on the break here. We're running behind. So I'm going to get started. Folks are okay with that. So my name is Dylan Tisdall. I am a research fellow here at the Martino Center, working in the LCN. And most of my work is on pulse sequence development. But today I am going to give the non-physicist intro to MRI, which hopefully means that it sounds more approachable and friendly. I am not a physicist. The bad news is I am a computer scientist and mathematician. I'm not sure whether that means that this talk is going to be a whole heck of a lot clearer. But here we go. So the first part of the key part of an MRI scanner is the fact that it is a really big magnet, a really big, really strong magnet. And I show this because you probably all remember this experiment that you get to do at some point in school where you drop iron filings on a piece of paper with a bar magnet underneath, and what you end up seeing is the field lines, right? The iron wants to align along the lines of the magnetic field. And if you were to sort of imagine what would happen if you did this experiment in an MRI scanner, please don't. Ideally, you would see that the field lines are almost perfectly parallel, pointing down the center of the bore. So down the center of the hole, the center of the magnet. Right? The field lines are almost perfectly parallel. And so that is actually a very hard engineering challenge, and that's part of why an MRI scanner is so ridiculously expensive. Your goal is to make a really big, really homogeneous magnetic field, all aligned, pointing down the bore. And we are going to call that, well, most of the time in a scanner you call that Z, X, Y, Z. But that's okay. Down the bore will suffice for most of what we're going to talk about today. So the other thing you need other than a magnet is a human head. And a fishbowl is a very good stand-in for a human head from the point of view of MRI. And basically what we're interested in MRI is the fact that you are all dirty bags of water. I love you all very much. But you are mostly water with some protein floating in it. Right? And what we are interested in is the interactions between the water molecules and the proteins, the macromolecules that make up your tissue. And it's those interactions that make it interesting. So from our point of view, it is some stuff floating in water. And just to remember, water is two hydrogens and an oxygen. And the interesting part of what happens in the water molecule is that the electrons are very strongly attracted to the oxygen nucleus. And so you end up with these sorts of, the protons in hydrogen are particularly nice for the MRI experiment. And I'm not going to get into the physics of why, because frankly, it's not my specialty either. But basically what we are doing most of the time when we are doing MRI on humans is we're imaging protons, right? And that is those two protons in the hydrogen atoms in the water molecule. Now, you can image things other than protons, and you can image things with different resonances and all sorts of exciting stuff, and it requires different coils and all sorts of fanciness. So most of the time when people come to the FreeSurfer talk, I'm pretty confident you're not doing sodium imaging or exciting stuff like that. If you are, that's great. That's not what this talk is about. This is about water and magnets. So if I take my water molecules, if I take my sample and I stick it in my big magnetic field, and I'm going to reduce this all down to 2 dimensions because it's far easier for me to draw. So I'm going to imagine that the main magnetic field is pointing, well, up here, which would be the same thing as down the bore, so through the center of the hole. What seems to happen when you do this is that everything, all these little water molecules, they are essentially tiny little magnets. Right? Those protons are essentially tiny little magnets, you can think of them as, and they will align with the magnetic field. They will align in the direction. Now, in reality, it's a little more complicated. They align either with or against it, and on average, a few more of them align along the field, but because there is so much water, in terms of the number of molecules that are actually there in any given voxel size you would care to measure, we can sort of approximate it by saying, oh, okay, well, we'll just pretend that they all aligned, preferentially along the field. It makes for an easier way to think about it. And then what do we do in our experiments? Well, you hear these terms like pulse. So we pulse them, and that causes them to flip away from the main magnetic field. So they rotate away from the main magnetic field when we add energy to them, right? So when you talk about pulsing or exciting the tissue, that's what you're doing. And then when we talk about flip angles, what that really is the angle away from the main magnetic field that we have rotated into the pulse. So these are all common terms that you hear when you're talking about MRI pulse sequences, right? You hear about the TR, which is the time between the pulses. You hear about the flip angle, which is how much you're going to be rotating it. So hopefully this is at least a little bit of a conceptual model for what's going on. Now the interesting part is that when you tip them over, you get this interesting effect where they start to spin. And they spin kind of like a top. And this is the classic way of describing this, and then I always wonder who actually remembers what happens when you play with a top, unless you've got small kids at home or something. But just to remind you, right, a top spins very quickly around its main long axis. But if you push it over, it develops a secondary wobble, which is kind of interesting. It develops a far slower wobble, and that is called what we call precession. And I'm going to show you a little diagram here that hopefully makes all of this a little bit easier. So this is going to be a movie. I'm going to describe it first because it's a little bit fast, and then we'll watch it. So basically what's going to happen here is we can think of the white arrow as all the aligned magnetization of the water. So that's the aligned protons. And then that yellow line that I'm showing you there is the pulse that I'm applying. It's the direction of the pulse. Right? And so the pulse I'm going to apply, so if you remember, they're all aligned along Z, right? Pointing down the bore. Here is up. And I'm going to apply this pulse along Y. The one thing that's maybe a little confusing to folks at first is when you apply the pulse, it rotates around the pulse. It's like it's another magnetic field that it's rotating around. So in this case, if I apply a pulse on Y, that white arrow is going to rotate down towards X. All right? So this is cool. So now we'll just make it happen here. There. So as you can see, if I pulse, then what happens is it starts to develop this secondary wobble. So you can think of it as if it was a top, it would be spinning very fast along its narrow long axis, like spinning around the point of the arrow. But then when it tips over, it also gets this secondary wobble. All right. So the key feature is that the rate of precession changes linearly with the strength of the magnetic field. So when you go from 1.5T to 3T, they're precessing, spinning twice as fast. So we have these spinning protons, or spinning magnetic moments, and how do we get any signal out of this? Well, if you think back to probably another experiment that you did once upon a time, where you wrap some copper wire in a coil and move a magnet through that coil, what that ends up doing is inducing a voltage in the coil, right? We get a measurable signal. If you move a magnet through the copper windings. And that's essentially what we're doing in MR here, is these little spinning protons are our moving magnets, and we wrap them in a coil, which is what our head coil is. And so it's the spinning, precession of those magnetic moments that generates the signal that we're going to measure in the head coil. So if I go back to this movie, the signal strength that I'm measuring, you can sort of think of as the length of the shadow cast onto the floor of this diagram. Right? There has to be something that's moving in the XY plane for me to get any signal in the coil. As long as it's just pointing along Z, as long as the magnetic moments are just pointing along Z, I don't measure anything. I measure when they're precessing, when they're spinning, and how much signal I get is related to the length. And you can see probably that, so what I'm measuring now is that little shadow that's cast along the bottom here. Right? The key feature that I'm sure you can guess is that the further over I flip it, the longer that shadow is going to be, the more signal I'm going to get, right? So until I get to 90 degrees. And if I go past 90 degrees, then that shadow is going to start looking smaller again, right? So I measure the amount of magnetization that is pointing in the XY direction, not the amount that's left in Z. Okay. Now, it's far easier to think about the remaining parts of what we're going to talk about here if we forget about precession for a second, or I shouldn't say forget about it, but we think about things as if we're in a frame of reference that's rotating with the precession. And the way I like to think about that is if you think about a merry-go-round, if you're standing off the merry-go-round, then everything on it looks like it's spinning. If you're standing on the merry-go-round, then everything on the merry-go-round with you looks like it's still, and the outside roll looks like it's spinning. Right? So we are going to get on the merry-go-round, and so now we are going to be spinning at the rate of precession. And so things that are precessing are going to look like they're still. Right? And I guess you can think about the MR scanner spinning around it at some crazy speed, but that's not probably that important. All right, so the next thing is once you pulse these magnetic moments and tip them over, they don't just stay that way forever, right? It doesn't just stay there and keep spinning. It gradually returns back to the original state of being aligned along the main magnetic field, and that process is called relaxation. So you can think of that as these things gradually realigning, right? So we put this energy in with a pulse, they relax, return the energy back out. So there's two different kinds of relaxation processes, and they're both really interesting in terms of describing tissue, which is why MR is so exciting. So when we tip them, they just rotate, right? They rotate towards the XY plane from Z. But when they relax, there's actually two different processes, and it is not just a straight rotation back upwards. So as much as I just drew it that way, that's me cheating. It's not actually like that. So there's two parts. We're going to deal with the first one first, the second one first. So T2 is, you can think of it as dephasing, maybe. And I'm going to describe what that means. So when I tip them over, things that are spinning a little bit faster, remember I'm in the rotating frame of reference now, things that spin a little bit faster than me end up out of phase, and they look like they're moving away from the main magnetization, right? Where things that are processing a little bit slower, for whatever reason, end up falling behind. And as I let this experiment run for longer and longer and longer, right, the things that are all these magnetic moments are processing at slightly different rates. And so they all end up out of sync with each other, right? And so over time I end up with something like this, where if there's all these little water molecules, because they're all processing at slightly different rates, when I first flip them over, they're all perfectly in sync. I get one long shadow along the floor, right? But if I let them run for a while, now I look at the signal I'm measuring. I look at the shadow on the floor. It's the sum of all of these arrows all pointing off in different directions, right? And of course, we know what that means, that they all cancel out, right? If I just do the vector addition of what is the length of the average shadow on that floor, it's going to be pretty close to 0, right? Because they're all pointing off in different directions. So dephasing looks, if I take the sum of all those little arrows that I just showed you, then this T2 effect looks like, right? We're going to see the T2 effect, we're going to see it on the floor. It just looks like the signal's going away, right? Because I don't get to measure each one of those little arrows, I only get to measure the average of them because they're all wrapped in my coil, right? So my coil just measures the sum of all the stuff in there. And so as they end up out of phase, it just looks like my signal is disappearing. So T2 is this process by which the water molecules end up out of phase with each other. Okay? And that's one relaxation process. And the other one, you might be able to guess the other component of this, is T1, which is the rate at which this arrow, right, these magnetic moments, lengthen back along the Z direction. Right? So we've said it gets shorter in X and Y because of dephasing. Right? So I end up not being able to measure any signal length there. We know it has to return to pointing along Z, so the other component of relaxation is the part with which it's slowly, the magnetic moments slowly return in Z. And so if I plot both of these things together, simultaneously, both of these processes, the way they happen in reality is I'm going to see T2 as shortening along the floor. I'm going to see T1 on this wall over on the side, right? Because I'm going to watch lengthening of the shadow there as my magnetic moment gets long again. And so I'm going to see something like that, right? Where I flipped it over, and now it's gradually relaxing back, right? And so at the end of this process, you can see there's almost no signal left in the XY plane, right? The shadow down at the bottom is essentially back at 0. And the shadow along the wall is now essentially back at full length, right? And so that's a full relaxation process. I flipped it over, it was processing, I could measure the signal for a while, gradually that signal decays away, and now I'm left back in the original state again. So I guess one of the sort of interesting parts about all of this is that you can go through this experiment where you pulse the subject, have them go through this complicated relaxation process, and it really doesn't seem to have much impact on our chemistry, in the sense that this is, as far as we can tell, other than the heating component that goes along with putting this energy into someone, we're not encoding specific biological information in the state of those spins. But what is neat is that as the water molecules interact with the proteins, as they interact with the molecules that make up the rest of your tissue, it dramatically changes their T1 and T2 properties. And so what we end up finding is that different tissue types, the water in different regions of your brain, or any of your tissues, have very different relaxation times, very different T1s and very different T2s. And those differences in relaxation are what allow you to get spatially or tissue-dependent signals out of MR. It's why gray matter and white matter give you different signals, is because they relax differently. And that's a really sort of neat accident that we're able to measure stuff this way. So in the metaphor of the fishbowl, it's the fish that make it interesting, right? If it was just pure water, we would just get one boring signal out of it. The reason we get an interesting signal out of our sample is because of the interactions between the water and the other molecules that are in there. And so one great way we take advantage of that, and hopefully people here know the MP-RAGE or IRS-PGR sequence if you use GE, I forget what Phillips calls it. But this is sort of the standard sequence that FreeSurfer takes in order to do its morphometry, right? And this sequence has nice gray matter, white matter contrast. And the way it achieves that is using a trick called inversion recovery. And so imagine for a second that what I'm showing you right now, again, right, I've got my pulse that I'm going to apply, only this time instead of a 90-degree pulse, right, up till now I've been flipping them 90 degrees. This time I'm going to flip it 180 degrees to start with. I'm going to turn it all the way upside down, right? And so there's no signal I can measure from that to start with, right? I turn it 180 degrees. And so if we think about what's going to happen, well, T2 will make it shorter in the XY plane, but there's no signal in the XY plane. So my T2 effect is negligible. But T1 is going to make that arrow want to grow back along the positive Z-axis again, right? So I'm going to turn it all the way upside down, and then it's going to want to return to being in positive Z. But if different tissue types have different rates of doing that, then they will end up out of sync with each other and returning back towards the Z-axis. And there will be some point at which white matter, for example, has crossed over that null point right in the center, but gray matter will not have crossed over, for example. I may have those backwards in terms of the relaxation rates, but you can understand that gray and white are going to relax at different rates. And that allows me to do this clever trick where I can wait until one of the tissue types is right at the null point, and the other one has some remaining magnetization that is either positive or negative above the Z, and then I can apply a 90-degree pulse, and that will flip it into the plane where I can measure it. So let's just watch that process here, now that I've talked you all through it. So we're going to do a 180 pulse. You can see there's two different types here that are relaxing at different rates, and when we get to the point where the second one is null, bang, I hit it with a 90. So now what am I measuring? I'm measuring the shadow on the floor, but only one of those two components has any contribution to that shadow. The other one was right at its null point at the moment I hit that. So let me just show you that one more time. Right? So there's two arrows, they're both the same length, they're superimposed on top of each other. I hit them with a 180, they have different T1 relaxation rates. I wait till the second one is null, and then I hit it with a 90, and that means that I can, by measuring the length of the shadow on the floor, I get just the contribution from one of those two tissue types. And that whole idea of inversion recovery, of picking what's called the inversion time, the TI, you do a 180 pulse, you wait some period of time, and then you hit it with a 90 or some other kind of, or some other smaller pulse, and you measure the residual component. By changing the inversion time, you can see you would get a different mixture of those two components, depending on how much they've each been allowed to relax. And that is a key feature of how you get good gray-white contrast out of an MP-RAGE, or lots of other sequences that use inversion. Yes, measure blank on the bottom. So using inversion recovery, we can weight our measurements for tissues with specific T1s. Yes?
When the second tissue is the magnetization is zero, it means that the amount of the speeds which are in parallel and anti-parallel positions are the same. Upon receiving the pulse, some of the anti-parallel ones will go to the parallel, and some of the parallel ones will go to the anti-parallel. But there are still some speeds in anti-parallel. So why on relaxation these anti-parallel ones do not contribute to the signal that we're receiving, because they will relax?
So you're saying, to make sure I understand your question, So you're saying when I apply a 180 pulse,
Yeah.
some of them are now, so I've reversed their orientations, right? The net magnetization before, if you imagine that, 51% of them are aligned along positive Z and 49% of them are aligned along negative Z.
Yes.
When I apply a 180, I've reversed those.
I mean, the second tissue having the net magnetization of 0, the amount of the parallel and antiparallel are exactly the same, both 50%.
I mean, that's one way to think of it. Yeah, I mean, so everything I'm doing here is dealing in approximations, but sure, yeah. Mm-hmm.
Then upon so the one that's having net magnetization of 0 has the same amount of
Yep.
antiparallel. Then upon receiving the pulse, some of them just change, some of the parallels go antiparallel and vice versa. And now we are having still 50% antiparallel. So upon
Well, if I apply a 90-degree pulse to something that's 50-50, positive and negative, and I tip that into the 90 plane, it still cancels itself out. So the question, right? So if I take this set of vectors that are 50% positive and 50% negative, and I rotate them 90 degrees, their sum is still 0. And so if I apply a 90-degree pulse, they're all going to rotate around that pulse. Right? Now, admittedly, when you apply a pulse, there's actually more complicated chaotic behavior that goes on. I apply a 180 pulse, and a very few of them actually rotate 90 degrees, and some of them rotate 180. The actual spin dynamics of this stuff is far more complicated, and that's why making pulse sequences is hard. But to a first approximation, that's the basic idea of inversion recovery. But yeah, you're right, that there is a cancellation that you have to be paying attention to. And we're just sort of summing it all up, and so it looks like it's disappeared. But obviously we haven't gotten rid of the energy. We didn't just make it go away. Okay, so that's a nice example of how you get contrast. But the next issue you want to think about is how you get any spatial information, right? MR gives you these beautifully high-resolution images. And so far, all we've talked about doing is measuring the sum total of everything inside the head coil, right? Which would be great if you wanted to do spectrometry. But no, if you wanted a spectra, great. But if you want an image, that sucks. That's one voxel. So how do we get information, right? So when we measure, you can think of what we're doing here. So now I'm going to take that 3D diagram I was showing you where I said I'm adding up the shadows, right? And here, if we turn this back into 2D, what are the shadows? The shadows are the length of those little red lines, right? And so I stick my whole sample in my head coil, right? The whole head goes in. And what does the head coil measure? It measures the magnetization that's processing in the XY plane, and that's the sum of all those red lines. So again, that's great if I want a 1 voxel image. So I'm going to walk you through how we turn this 1 voxel measurement into a 2 voxel image. So imagine I want 2 voxels, left and right. So the first thing I do is I pulse and I take my first measurement. And that first measurement is the sum of all the magnetization that's in there. Now, imagine I can apply a different magnetic field to each half of the sample, right? I've got these things called gradients, right? We know MR standards have gradients, X, Y, and Z, right? And so what the gradient allows me to do is apply a slightly weaker magnetic field on the right side and a slightly stronger magnetic field on the left side. Backwards, left side, right side, but you can tell. Right? And as we said, the rate of precession is linear in the magnetic field, right? So if I make it a little bit weaker, they're going to spin a little bit slower. And if I make the field a little bit larger, they're going to spin a little bit faster. So what I've done effectively by applying this gradient is I've made the rate of precession different in my 2 different voxels, right? And so if I wait for a minute, far less than a minute actually, milliseconds, then they will end up perfectly out of phase with each other, right? I know how strong the magnetic field I'm applying at each point is. I know how fast they precess. So I know exactly how much time I need to get the 2 halves of my water sample out of phase with each other. So now, I wait until the voxels are perfectly out of phase, and now I take a second measurement. And I sum over everything in the fishbowl. But you can see now there's cancellation, right? This is the sum of the stuff on the left minus the stuff on the right is my measurement, right? So I don't get to choose to just measure 1/2, but I can build up cancellation patterns by applying gradients and waiting set amounts of time. So let's think about this. Our first measurement was the sum of the left and the right. Our second measurement was the left minus the right. So if I add the two measurements together, I get 2 times the left voxels intensity. And if I subtract them, I get 2 times the right voxels intensity. So therefore I can make an image out of this. I can make a 2 voxel image, which is all I was promising, by taking these 2 measurements and using the fact that they have different cancellation patterns to figure out how much signal came from just the left part and how much signal came from just the right part. And I can turn that into an image. So real sequences are going to sum together fractional amounts from all the voxels. So here I was doing a very crude approximation to this. But in the real world, you end up with little fractional bits from all the voxels. Also, you do it in 3 dimensions. You do it in X, Y, and Z. And then the voxels are unmixed, so instead of the very simple cancellation pattern I had here, you can work through the mathematics, and there's a transform called the inverse Fourier transform that you can prove allows you to invert this fractional mixture that you get by applying the gradients for different amounts of time. And so by doing that, you can end up with the spatial information you need reconstructed, even though you only are ever able to measure the sum over all the signal in the head at any given moment. You can unmix it by creating these cancellation patterns with the gradients. All right. So then a pulse sequence you can think of as prepare the signal, so you can invert it, wait some period of time. You can flip it with different flip angles to get more or less signal. And then you want to localize the signal using the gradients. Then you want to take a measurement. Then you want to let the tissues relax. And you want to go back to step 1. Right? You're going to repeat this maybe for every voxel. And if you do that and you work through the math, it's going to take you a couple of days, and your subject is not going to stay in the scanner that long. So that's a bad idea, right? Relaxation, T1 of brain tissue is on the order of seconds. Right? So it's a long time if I've got 256 by 256 by 256 volume that I want to measure. Not going to fly. So all the fanciness in pulse sequences, all the actual tricks, are in figuring out how to localize and measure repeatedly after one flip and before the tissues are able to completely relax. So when you hear about pulse sequences like flash or EPI or any of those sorts of things, what those are different techniques for accelerating what is otherwise a terribly slow experiment. And so there are ways that you get multiple steps of spatial localization information out of one excitation relaxation pattern. And so you can get your experiment down from days to minutes. Okay, so then 2 other quick topics. First of all is bold FMRI. How does this work? This is where I completely push my fishbowl metaphor to the point of being ridiculous. So we can imagine I've got 2 voxels, right? They're processing at the same rate. They're in phase. I flip them over. This is good, right? I tip them. They process. Excellent. I add a source of iron. Might be blood, or it could be a fishhook. And what's going to happen is that now I've changed the magnetic field in that side. I have caused local variations in the magnetic field right around the fishhook, because the fishhook is magnetic as well. I don't have to make it magnetic. I don't have to, but the magnetic field is distorted by it. It has different susceptibility. And so what's going to happen is that the side of the fishhook is going to dephase faster. And it's going to dephase faster because every one of those little water molecules is in a slightly different magnetic field, so they're not in sync anymore, right? And so as they dephase, right, I get less signal. Remember, T2 is dephasing, right? So if I speed that process up, then the left voxel, when I take a measurement, is going to end up with less signal than the right voxel, right? Because all the water in there is not able to stay in sync. And then when I pull the iron back out, right, if I take another measurement, then the T2 returns to normal, and both voxels will have similar signal intensities again. And so basically what you're measuring when you're measuring the bold effect is you're measuring these temporal changes, the variation through time in dephasing that's caused by, or the variation in the magnetic field that's caused by the inflow of blood. Right? That's the core concept here, but really what you're measuring is the way it changes T2. All right, the other simple one I want to talk about is diffusion. So everyone loves diffusion experiments. So just to quickly talk about how they work, we're going to simplify this down for a second, and we're going to imagine that we only have two of these. And we can see, right, if I apply a gradient, then they're going to end up out of phase with each other. Right? I apply a gradient that is, this time I'll get it right, weaker on the left, stronger on the right. Right? And so I apply that for a while, they end up out of phase. Now I leave no gradient. I turn the gradient off. Right? And they will just keep processing, now at the same frequency again, and they'll stay out of phase. Now I apply the opposite gradient for the same amount of time. And they will end up back in phase. Right? Because now the one that was spinning faster before is now spinning a little bit slower, and vice versa, so they catch up with each other as long as I apply the same gradient with reversed polarity for the same amount of time. Right? I can unwind the same effect. So that's great. Stationary things, I can do this trick where I turn on a gradient, dephase them, play the opposite gradient, rephase them. Good. Water molecules, though, we know are not stationary. Right? In human tissue, they're diffusing. You expect over, say, 50 milliseconds that they're going to move about 17 microns just due to diffusion. That's not due to, flow, like blood pumping or any of that sort of stuff. Just at that temperature, the free diffusion of water. You expect a significant motion. And so now we're going to go back to 4 little sample molecules here. I'm going to think about what happens if they move while I do this experiment. So first of all, I turn on the gradients. The ones on the left end up out of phase with the ones on the right. I turn off the gradient, so they're just relaxing. But now, a couple of them are going to diffuse. Right? So we're going to ask, but you note that they are diffusing in the direction I was varying the gradient. So I was varying the gradient horizontally, right? Things on the left saw something different than things on the right. So if water is diffusing left to right, then when I put the gradient back on, I'm not going to be able to unwind that cancellation effect, right? Because things experienced one gradient where they were wound up and then they moved to a different position and now they don't get unwound. So the stationary ones, yeah, they got refocused. But the ones that moved, the ones that diffused, aren't refocused. And so the signal has canceled out because there was diffusion parallel to the gradient. So if I apply those diffusion gradients, now I don't measure as much signal. In the same way in the bold experiment, if there was blood flowing in, then I didn't measure as much signal. Here, if water has diffused in the direction that I was applying the gradients, even though I apply them and unwind them, anything that's moved in that direction doesn't get, the signal doesn't come back. So let's just think about what happens if the direction of diffusion is perpendicular to the gradients. And this is why the diffusion experiment is so interesting. So again, I'm going to apply the gradients. Right, left, right, out of phase. Yep, we got this. No gradients. Yep, everything stays in sync. Now we're going to move them perpendicular to the direction of the gradients. Now when I apply the opposite gradient, everything ends up refocused again, right? Because even though they moved, they didn't move in a way that changed the gradients they're going to experience in the 2 parts of my experiment. And so the stationary ones were refocused, and the diffused ones were refocused. So the signal is unaffected by diffusion perpendicular to the gradient. So the key point that I want to drive home here is diffusion imaging uses the gradients to cancel a signal in water that moves in one direction. And that's why you need a whole bunch of different diffusion directions. You have to measure them in order to see the different ways that water might be diffusing. So you repeat the experiment each time using a gradient in a different direction, and that gives you a map of how freely water is diffusing in each voxel. Right? In directions where the water is diffusing freely, you'll see lots of cancellation. In directions where the water isn't free to diffuse, if there's no motion in that direction and you apply a gradient there, then there'll be no signal cancellation. And so you can fit the changes in signal intensity as you vary the encoding direction in order to model or map how much diffusion there is. All right. And with that, questions? I know that's a whirlwind tour through it, but hopefully it gives a little bit of intuition about the different ways that contrast and spatial information are had in the MR experiment. Everyone is stunned. All right.